So are you all refreshed? I'll get your seminar first draft done. Final draft? Okay. Reason I added 2 points on to everybody's exam is I was looking through the question. There were really 2 questions that tested the same concept, which a lot of people missed. That was the concept of identification. And so I didn't want to penalize you twice for not knowing that concept, so that was why I added the 2 points on. Class distribution up here, and then past history, and you can see that it's really pretty consistent from year to year what the performance on the second exam is. So you're right, exactly what last year's was, a little bit lower, but not any significant difference with the last 3 years. Exams tend to get a little bit harder as we go along because the material gets a little bit more complex. That's particularly going to be true with the last exam, and that's one of the reasons why we don't have a comprehensive exam in here, because all of this stuff about the immune system and immune function is really new for you. So try to just do your best to keep up. That's the best advice that I can give you. Hopefully you'll be able to make up some points on the lab exam, because the lab exam really should be pretty straightforward. If you've been doing the work in lab, particularly this first lab exam where it's a lot of hands-on things, then grade should be a little bit higher. So, questions? Okay, no.
Just to be sure, you said we come in at 8:30 on Wednesday, right?
Yeah, now that we had a couple of people listening, so I would like some people to come in at 8, actually, if you can. So how many of you think you can come in at 8? On Wednesday? We probably, let's see, yeah, that's fine. And then the rest of you, if you could maybe just stagger by 8:30. If you happen to, don't have an 11 o'clock class, maybe come in at 9 or so. Sure, that's fine. I'm just trying to prevent a rush at 9 o'clock where everybody's in here at the same time trying to get to all of the stations. I will be having to close up the lab on Tuesday afternoon to set up the lab exam. So probably at noon, the lab will be closed. If you want to, I can leave the little research lab across from the elevator open if you want to bring your microscope in there and put some slides in there. So I'll give you a reminder tomorrow that the lab will close at noon. It takes me a while to set up the exam, so you won't have access to it Tuesday afternoon or obviously Tuesday night. Okay. Okay, let's take a couple of steps back and do a little bit of review, because the material we had right before the exam, you were probably a little bit distracted. Then break, and so you've probably forgotten pretty much anything that we talked about that day before class started. So what we're doing is we're going through some of the drugs that are responsible for selective toxicity, killing a pathogen, but hopefully not harming the host. Although we're going to see later that even good drugs typically will have adverse side effects, particularly if they're not dosed properly. At some point, any drug can become toxic if you exceed certain recommended dosing levels. And so you've always got to be careful. But if these particular antibacterial drugs can exploit a difference in the physiology or anatomy of the pathogen compared to the host, such as cell wall structure or protein synthesis mechanisms, then at least you've got a fairly selective mechanism to try to kill the pathogen and not harm the host. We talked about penicillins kind of historically and their discovery. The mechanism of penicillin function is that it ultimately contributes to cell lysis, particularly if the organism is in a hypotonic environment. because then the cells will take up water, and with the loosened cell wall structure, eventually those cells will lyse and burst. And so I showed you these pictures of cells in the presence and absence of penicillin, and down here at the bottom you can see several cells which have had weakened cell walls, which have now lysed. Some of these cells have not been affected yet, or maybe won't be affected at all. Some of these might be resistant to that particular antibiotic. The major mode of action is that these drugs, these penicillins, of which there are several types, will bind to bacterial enzymes and those bacterial enzymes are referred to as penicillin binding proteins, or PBPs. So what are these enzymes? Well, these are enzymes that are involved in peptidoglycan biosynthesis. There are two enzymes in particular that penicillins bind to quite readily. And this binding is a competitive inhibition. So the penicillins are going to look something like the substrates that these enzymes normally bind to, and if the enzymes become bound up with these inhibitors, then they won't work as readily on their normal substrate. Two of the major penicillin binding proteins are the proteins transpeptidase and transglycosylase. I showed you this previously, but let's go back and look at it again. Remember that I made the statement that in order to understand how these penicillins work, you really have to understand how peptidoglycan biosynthesis takes place. And it's not a necessarily straightforward process. I'm going to abbreviate it a little bit for you, okay? So this particular diagram right here kind of summarizes some of the key steps involved in peptidoglycan biosynthesis, okay? Remember what peptidoglycan is. Peptidoglycan is a polymer of sugars. The two sugars are NAG and NAM, and they're hooked together in this chain. And then NAM sugars additionally have this group of 5 amino acids that are attached onto it. And those 5 amino acids can link with other chains of 5 amino acids within that peptidoglycan strand to strengthen it. Okay? So, this dotted line represents the cell membrane. This is what's happening in the cytoplasm. And then this is what is happening out at the cell surface. Okay? So in a gram-negative bacterium, this would be happening in the periplasmic space. Remember, that's that space between the cell membrane and the peptidoglycan layer. If this were a gram-positive bacterium, then this would just be happening out on the surface of the cell wall. So basically what's happening is that peptidoglycan subunits are originally partially put together and joined together in the cytoplasm. Then they have to be transported across the membrane, and then these chains of peptidoglycan are assembled, and then these cross-links of these peptide units are joined together to give that further strength and integrity. So there's several enzymes that are involved in this biosynthesis process, and several of these enzymes are targets for inhibition by penicillin molecules and their derivatives. Okay, we mentioned that penicillin molecules are a type of antibody called a beta-lactam antibiotic. And beta-lactam antibiotics have this particular structure. There's this beta-lactam ring, and there's this thiazolidine. So this is kind of the basic structure of a penicillin molecule. Now each type of penicillin, we'll see in a minute, has some variations of molecules attached to this ring. But this is kind of the guts of the penicillin molecule. Okay? Penicillin is actually formed by joining two amino acids together during a fermentation process. Cysteine and valine are hooked together to form this beta-lactam structure. Okay, keep that in mind for just a second. Okay. One of the key events in the synthesis of these peptidoglycan subunits is that N-acetylglucosamine, one of the two sugars, has 3 amino acids attached onto it, and then 2 alanines are attached to those 3 amino acids. That gives you what we call this NAM pentapeptide, and that's represented just by the NAM of 5 little circles, representing 5 amino acids. But the latter 2 here are these 2 alanines. Okay? This is going to be a real target for inhibition right here. Because as we'll see in a minute, these alanine-alanine dipeptides are recognized by some of these biosynthetic enzymes in the process of making peptidoglycan. This is the substrate that the enzyme recognizes. Okay? Well, here's the structure of an alanine-alanine dipeptide. Here's the structure of the beta-lactam nucleus of penicillins. They look a little bit the same. What's going to happen is that if you feed bacteria this drug, this penicillin drug, that it's going to mistake the drug for the alanine-alanine dipeptide. And these enzymes that normally bind that and synthesize peptidoglycan are going to be fooled into binding the penicillins instead. And that's going to competitively inhibit those enzymes. Okay? If there's way more way more penicillin than there is normal alanine-alanine substrate, then those enzymes are going to get tied up by that inhibitor. And therefore it will disrupt peptidoglycan biosynthesis. So for example, one of the first steps in the synthesis of this pentapeptide is a couple of alanines are stuck together to these 3 amino acids forming the pentapeptide. There actually is an enzyme that catalyzes this reaction, and there's an antibiotic that inhibits that enzyme. The antibiotic is called cycloserine. That's not what we're going to talk about. Once we've got this NAM pentapeptide, then that attaches to a carrier molecule in the membrane. It's called a bacropanol. And that carrier then carries that NAM pentapeptide up to the membrane. N-acetylglucosamine is added to that. So now we've got 2 sugars with the peptide branch sticking off of it. The carrier flips, and now that is transported out of the cell, out to the surface. So we've got this NAG, NAM, with the pentapeptide attached to the NAM. Okay? Well, here's our 2 enzymes, transglycosylase and transpeptidase. Transglycosylase adds these 2 sugars onto an existing peptidoglycan chain. It lengthens it. Transpeptidase then hooks the 2 peptide bridges together. This one was here previously. This is the pentapeptide that was added. So this enzyme now stitches those 2 together to strengthen the peptidoglycan molecule. Okay? Well, these enzymes, these penicillin-binding proteins, transglycosylase and transpeptidase, are recognizing this alanine-alanine bridge right here. And penicillin looks enough like it that it inhibits those enzymes. So there's competitive inhibition of those penicillin-binding proteins, or actually bacterial enzymes that are involved in penicillin biosynthesis. Okay. So understanding how this works biochemically then gives us some knowledge that allows us to create drugs which will competitively inhibit this particular process. Now, we're going to come back later on and talk about a couple of other antibiotics that are not penicillin-type molecules. And they're going to, as it turns out, inhibit this process in a little bit different way at a little bit different step. One of the examples we're going to talk about is the antibiotic vancomycin. Vancomycin is really important because as bacteria have become resistant to the penicillins, they haven't become yet resistant a lot to this vancomycin because it has a different mode of action. And so vancomycin is kind of one of those last reserve drugs that are used if you have an infection and it is a penicillin-resistant form of that infectious agent. Then we use vancomycin. But the more we use vancomycin, the more that organisms are going to evolve resistance to that particular antibiotic. Okay. So, here's penicillin. This is a naturally occurring form of penicillin, sodium penicillin G. And this is another naturally occurring form of penicillin, penicillin B. These are the two types of antibiotics that are naturally produced by the fungus that Fleming first discovered back in the 1920s. And now we know what they do. Okay, and here's the key structural element right here. This beta-lactam nuclease with this beta-lactam ring and the thiazolidine ring. Looks a lot like the alanine dipeptide. Okay. Now, naturally occurring penicillin G and V are considered to be narrow-spectrum antibiotics. If you're narrow-spectrum, then there are a limited group of organisms that you are effective against. And specifically, these particular antibiotics were primarily against gram-positive bacteria. Why? Because these molecules can't be transported across the LPS layer and get into the periplastic space where those enzymes are found that penicillin normally inhibits. So if the drug can't get access to these enzymes, it's not going to work. There are some other limitations of naturally occurring form of penicillins. They have very poor oral absorption. And they are not very acid resistant. So if you try to take these drugs orally, most of the penicillin would be degraded in the stomach to begin with. But even if some molecules survive, they would be poorly absorbed across the gut into the bloodstream. So instead, these have to be injected. Okay? So the first penicillins that were given were not a pill, they were a shot. Well, when drug companies started to produce these molecules and start selling them and trying to make profit, they said, wait a minute, it's not going to be nearly as convenient for people to take these drugs if they've got to be injected, because then you've got to go to the doctor's office. Or, back when doctors used to make house calls, they really did that. The doctor would come, open his black medical bag, usually was a key at that time, and give you an injection of penicillin. If you could make a synthetic penicillin that had good oral absorption and good acid resistance, then you would certainly increase your market share because people would be more likely to buy the drug and doctors would probably be more likely to prescribe it, particularly as they move toward this more office-based model as opposed to the home-based model of care. So, drug companies turned to organic chemists loose and said, we need to preserve this because this is really what's causing the competitive inhibition. Can you tinker around with this part of the molecule, okay, and see if you can alter it to overcome those limitations? And they did. They started to develop some semi-synthetic forms of penicillin in the lab. Okay, these are not naturally occurring molecules. These are molecules that are produced by tinkering around with the naturally occurring forms of penicillin. So, ampicillin, penicillin F, nafacillin, methicillin, aminopenicillic acid. Okay? All kinds of different modifications were done to try to overcome those particular limitations of the molecules. Ampicillin was one of the first broad-spectrum antibiotics. By tinkering around with this portion of the molecule, they were able to overcome the limitation that it could cross the LPS layer of gram-negative bacteria. And now this molecule was permeable to that LPS layer. Therefore, the molecule can now inhibit gram-negatives as well as gram-positives. I don't have a structure of this one, but how many of you have taken amoxicillin when you were young? Probably just about everybody. Kind of the go-to drug for ear infections, for example. Usually in this little pink liquid suspension. It's like the taste of it. Amoxicillin was broad spectrum, and it also was modified in such a way that it wasn't destroyed by the acid in your stomach, and also it had good oral absorption once it passed through the stomach. And so by fooling around with the chemistry of these particular drugs, you could really fine-tune them. Another thing that became important, and we'll elaborate on this later, is bacteria responded to the exposure to penicillin by becoming resistant to those particular drugs. We'll talk about mechanisms of resistance later on. But typically what happens is that there's a random mutation of a gene, and the gene results in a modification of a protein that can now do something different than it used to. Oftentimes that is preceded by a gene duplication. So you can retain the normal copy of the gene, but then the new copy of the gene can become mutated and gain a new function. One of the new functions was this enzyme could now bind to penicillin and degrade it and break it down. And that contributed to the penicillin resistance. Those particular enzymes are called penicillinase enzymes. So what did drug companies do? They went back to the organic chemist and said, let's retweak this molecule more and try to make it resistant to these newly evolving penicillinase enzymes. Methicillin was one of the first penicillinase-resistant forms of penicillin. And until recently, methicillin was the go-to drug for treating Staphylococcus aureus infections. What's happened now? Bacteria have responded by becoming resistant to methicillin. And now we have the MRSA, methicillin-resistant Staphylococcus aureus. We try to come up with a new alternative to treat the MRSA form of the bacteria. And what eventually are they going to do? They're going to evolve resistance to that. Okay? It really is natural selection in action. But more about that later on. So this particular slide gives you some kind of summary of various forms of penicillin and derivatives, talking about their spectrum of action, various uses and advantages, and disadvantages to this as well. So you'll notice, for example, that methicillin is a narrow spectrum antibiotic. It has pretty poor absorption, but it's not susceptible typically to penicillinases.
What makes some prone to allergies?
Typically, if you're allergic to one form of penicillin, you're allergic to all forms of penicillin because your immune system is responding to that beta-lactam nucleus, which they all share. But the substitute molecules on the beta-lactam nucleus can enhance that particular allergic reaction. For some reason, your antibodies that are causing those allergic reactions respond a little bit more strongly to certain molecules with certain R groups than other molecules with different groups. But they still share that same beta-lactam nucleus. If you look at amoxicillin, it's a broad-spectrum antibiotic. It's got good absorption, but a disadvantage is it is hydrolyzed by penicillinase. So typically it's hard to engineer a molecule that has all the advantages and none of the disadvantages. So doctors have to be careful, and they have to prescribe based upon knowledge of what the pathogen is. So you're not going to be quite as concerned about an antibiotic being narrow spectrum if you know the caused agent of the disease is gram-positive. Okay? So it's really important for doctors to do cultures so that they know the target that they're trying to kill. And unfortunately, that doesn't happen much anymore. Kind of go-to response is, I think it might be a bacterial infection, where very possibly maybe viral, and then you hit it with the broad-spectrum antibiotic. And it's hard to argue with that, because if you look at now about how doctors have to turn through patients through their office, they don't really have time to do all of the cultures. Tell the patient, well, you've got to come back in 48 hours and I'll have the result for you. No, patients aren't willing to do that. There is another type of beta-lactam antibiotic called cephalosporins. Cephalosporins account for about 1/3 of all of the antibiotic prescriptions today. It's becoming a very popular drug. These are also beta-lactam antibiotics. And consequently, if you are allergic to penicillins, you're probably going to be allergic to cephalosporins as well. Typically, you can tell that a drug is a cephalosporin because it has a name beginning with either Ceph or Kef. Ceplex is a pretty common cephalosporin antibiotic. If any of you have ever read Z-Pack, that is a cephalosporin antibiotic. Okay. The major difference between the cephalosporins and the penicillins is they both have this beta-lactam nucleus. In penicillins, there is one place on the nucleus where R groups are attached. Okay, so each one of these is different in a different penicillin. In cephalosporins, there are two places to which R groups are attached. So this gives us more variety of structure. Cephalosporins have gone through several modifications or generations. The first generations are the oldest ones that were produced. We are now in the 4th generation of cephalosporins. What's going on? What were the drug companies trying to do as they developed these new generations of cephalosporins? Well, basically, they were trying to improve their activity, and that typically involved increasing their spectrum to make them more broad-spectrum drugs, improving their oral absorption and their acid resistance. One of the things that happened particularly in the 3rd and 4th generations of cephalosporins is that the molecules were modified in such a way that they had better capability to cross that blood-brain barrier. Penicillins are very lousy at crossing the blood-brain barrier, and consequently are pretty ineffective against things like meningitis, where you actually get a bacterial infection of the meninges or the cerebral spinal fluid. You've got to cross the blood-brain barrier to cause that kind of an infection. The 4th generation and some of the 3rd generation of cephalosporins can cross that blood-brain barrier and therefore are effective against those particular types of diseases. So that was a real improvement. Another thing that they've tried to do as they've developed these new generations of cephalosporins is to try to minimize the side effects. One of the biggest side effects of any beta-lactam antibiotic is gastric distress. Okay, some people suffer nausea, some people diarrhea as a result of taking the antibiotics. And so by chemically modifying these particular antibiotic molecules, hopefully you can minimize those particular side effects. And again, If you've got two R groups you can tinker around with, you're probably going to have more opportunities to do that than the single R group that is found attached to the beta-lactam nucleus in penicillins. A couple of other antibiotics worth mentioning. One is the antibiotic bacitracin. This looks very different from penicillin. We don't have that beta-lactam nucleus. So this is probably going to have a different mode of action. This is not going to bind to those penicillin binding proteins and competitively inhibit it. It's going to do something else. You ever heard of bacitracin before? Put it on cuts. Okay, it's typically for just topical use. It's in the over-the-counter medication Neosporin, for example. So what does bacitracin do? Bacitracin inhibits this step right here in peptidoglycan biosynthesis. Remember that these NAM, NAG pentapeptides are transported across the membrane by that carrier, the bactropanol. And then they're cleaved off of that and added to peptidoglycan. And normally that bactropanol is recycled and moved back across the membrane so it can pick up more of these. Bacitracin inhibits the translocation of that carrier back across the membrane again. So it makes sense that this particular molecule has a different structure than penicillin. Here's vancomycin, the one that I talked about a little bit. This has got a very different structure than penicillin. And yet it inhibits peptidoglycan biosynthesis. How does it do it? What happens here is that vancomycin is binding to the pentapeptide itself. It's not binding to the penicillin binding proteins, like the penicillin's part, but it binds to the pentapeptide, this alanine-alanine unit. And that prevents the penicillin binding proteins from binding to the pentapeptide. You see the distinction there? And penicillins are binding directly to the enzymes. Here in vancomycin, they're binding to the peptidoglycan and preventing these enzymes from interacting with the peptidoglycan. Pardon me? I guess you could consider it that way, yeah. Well, this is good, because if organisms are evolving a resistance to penicillins by producing penicillinases, for example, they're targeting particularly the competitive inhibition of these particular enzymes by the penicillin. It's not how vancomycin works. And so the likelihood, because they have different mechanisms of them developing simultaneously resistance to vancomycin, is much lower. And so what? Well, we start using vancomycin now. And then they're going to develop their own strategies for dealing with this particular type of antibiotic. Here's another antibiotic you don't hear a lot about, isoniazid. This particular antibiotic is very useful in treating tuberculosis. Its mode of action is it blocks mycolic acid synthesis. And if the bacteria can't produce mycolic acid, it messes up its cell wall. Remember, mycolic acid is the waxy substance that gives you an acid-fast reaction in the acid-fast state. So this has a different mechanism of action than the penicillins or vancomycin or bacitracin. This is just the tip of the iceberg. There are hundreds of antibiotics that fall into this category of penicillin-related molecules or cell wall synthesis inhibitors. Another target that has been heavily exploited for antibiotics are the inhibition of protein synthesis. This is going to exploit the dependence of 70S ribosomes. Remember that the two subunits of 70S ribosomes are the larger 50S and the smaller 30S. Some drugs will interfere exclusively with 50S ribosomal subunits. Other drugs will influence or interfere with 30S ribosomal subunits. Just going to give you a few examples to give you an idea of kind of the variety of effects that these particular drugs can have. Here's an oldie but goodie. This is tetracycline. Tetracycline was one of the earlier protein synthesis inhibiting antibodies that was used and developed. When tetracycline was first approved for use, one of the first targets was sexually transmitted diseases, syphilis. And this had a real effect on society because prior to this, there really wasn't a good treatment for syphilis. Now that there was, a pill could cure you. And that really had an effect on public health standards. Public health standards previously have really been geared toward prevention. Now it's primarily treatment. If you get the disease, take a pill. The term tetracycline refers to the fact that this particular antibody has these 4 heterocyclic rings joined to one another. Tetracycline interferes with the 30S ribosomal subunit. And specifically, it interferes with the binding of the tRNA to the A site of that 30S ribosomal subunit. Well, if the tRNA can't bind to the A site, it can't then bring in its amino acid, and so this terminates protein synthesis. So the bacteria stop making proteins, and that obviously is evident.
It's like allosteric inhibition, going to not bind the active site of the
This is, I think this is really, Zach, more of a competitive inhibition. Okay, it really actually blocks the A site. Tetracycline has some downsides. This particular bacteria is fairly notorious for being nasty on your GI tract, so a lot of people respond badly to it. And it has an interesting side effect, and that is the tetracycline molecules tend to become deposited in hard tissue in the body, especially the bone. If pregnant mothers take tetracycline, it can become deposited in the bones and teeth of the developing fetus. If young children take it, it can be deposited in their enamel. And one of the things you can see, I'll show you a slide later on when we talk about side effects, is that sometimes people have mottled or grayish teeth because of the deposition of tetracycline in their hard tissues. That's one of the negative side effects of that one. Chloramphenicol is also a, no, sorry, that's a 50S. It interferes with protein synthesis on 50S ribosomal subunits. And it prevents peptide bond formation. Remember when the tRNAs are both in the P site and the A site, the peptide bond forms between the tRNA coming in and the previously joined together chain. So again, this will terminate protein synthesis. Quite lethal. Chloramphenicol is small enough, look at its small size compared to tetracycline, that this can cross the blood-brain barrier. So this would be effective in treating a disease like meningitis. However, it has a lot of toxic side effects. Chloramphenicol particularly is a very potent inhibitor of mitochondrial protein synthesis. Remember, mitochondria have 70S ribosomal subunits. And more so than other bacteria, this really seems to affect the bone marrow cells. That can result in a condition called aplastic anemia, where you get a dramatic decrease in your red blood cell production, and that can be life-threatening. So typically, chloramphenicol was used primarily for life-threatening conditions like meningitis. But as they've improved these third and fourth generations of cephalosporins, now chloramphenicol is hardly ever used anymore. They don't really have new generations, particularly of drugs like chloramphenicol. Erythromycin belongs to a class of antibiotics called the macrolide antibiotics. They're different because they have sugar molecules attached to the antibiotic. So here we've got this three-ring structure right here called a lactone ring. And then there are a couple of sugar molecules that are attached to that lactone ring. This is a 50S inhibitor also. This particular antibiotic interferes with translocation. Remember, during protein synthesis, that once the A and the P sites are filled, then the ribosome moves down 1 unit. Translocation. So this antibiotic is going to interfere with translocation. So again, terminating polypeptide synthesis. Erythromycin is a pretty handy antibiotic. Has much fewer GI disruption than tetracycline. It's certainly not life-threatening like chloramphenicol is. And so this was a fairly widely used antibiotic for a long period of time. It's still the drug of choice for the mycoplastic pneumonia, the walking pneumonia. Have you ever had erythromycin before? Is that it? What was What was it for? Pneumonias, yeah, it's still used quite a bit for pneumonias. Here's another type of antibiotic called streptomycin. This is also a sugar-containing antibiotic. Streptomycin interacts with 30S ribosomal subunits. And it does something kind of interesting. When this molecule binds to a 30S ribosomal subunit, it actually changes its shape. It's kind of an allosteric thing almost. Okay? And what that does is it distorts the message and causes the codons to be read incorrectly. So you make a protein, but it doesn't make any sense. So this makes a nonsense protein, which will then fold up improperly and not have the right function. Before the isoniazid was developed, this was one of the first antibiotics used against tuberculosis. Unfortunately, it had some side effects. This particular antibiotic seemed to be toxic to auditory neurons and resulted in deafness in a number of patients. A newer version of this that some of you may have heard of before, gentamicin, has a lot less toxicity than streptomycin does. These have been the major strategies for decades, inhibiting these two particular targets of bacterial cell wall synthesis or protein synthesis on 70S ribosomes. Within the last couple of decades, drug companies have tried to think outside the box and to try to develop some new strategies. Unfortunately, it takes a long time for those particular antibodies and antimicrobial drugs to be developed. I think we have had 3 new drugs with new novel actions that have been developed in the last 2 decades. So we're really falling behind in terms of this war of evolution of resistance and our ability to come up with new chemicals to which the microorganisms aren't resistant. So what we'll do a little bit next time is talk about some of these new strategies that are a little bit outside the box from the kind of things we've been talking about so far. If you have any questions about lab exam, tomorrow I guess would be a good time to get those out of the way. Okay? Is this what's wrong? Yeah, so that would, yeah, this is one a lot of people missed, and that's one of the reasons why I added on the 2 points. It's,
Yeah, because this was an old sentence. This was 2000.
yeah, what's wrong? Denitrification is anaerobic respiration. Yeah, I mean, a lot of people missed that question, and that was why the 2 points were added.
It was on both tests. They were not even.
And it was wrong in both tests?
Yeah.
Ah. Yeah, nitrification is the result of,
Huh?
Oh my God.
Get that
Real
out too.
bad?
Yeah, it's all good.
So I forgot so much.
oh, okay, okay.
Got to switch this one.
And I can sit right next to it.
Yeah, producing nitrate is the result of chemoautotrophy. That's where the ammonia is oxidized into nitrate. And then
Yeah.
denitrification is nitrate being
Which is anaerobic.
converted into nitrite, which is anaerobic respiration. Well, it's funny, I was wondering why so many people missed that. So,
Yeah.
I'm going to have to go back and change that.